Today I'm going to talk about sort of the tissue I've been studying in my postdoctoral work is the nasal mucosa. And so the nasal mucosa is a very exciting tissue to me as the primary site of viral infection. And Nancy, I think, already did a really good job of highlighting why we should be studying this tissue in general. And so what I've been interested in doing is asking a couple questions about this very important tissue in viral infection. So first, how does infection unfold in this tissue? And second, how do we prevent disease and infection from even starting in this tissue in the first place as the site of primary infection and the site of spread? And so we know a little bit about immunity in this tissue. We've known for a very long time that IgA can develop, the secreted antibody can develop in this tissue and is able to neutralize viral infection and can be boosted either by local vaccination or by subsequent infection. And also more recently, it's been described that we can have tissue-resident memory T cells that can develop in this tissue and be sustained and exhibit cytotoxic and activation signaling capacities. But more so than just sort of typical adaptive immunity, it's becoming more clear that the state of the nasal mucosa at the time of infection can impact disease trajectory. And a really nice study in the Gambia showed that children who were inoculated with live attenuated influenza vaccine, those who had seropositivity or qPCR positivity for other viral infections or bacterial infections exhibited reduced ongoing infection and reduced levels of viral shedding when inoculated with a live attenuated influenza virus, suggesting that recent infection predisposes the nasal mucosa to restrict subsequent infection. And also we've shown in some older work looking at SARS-CoV-2 infection that severe individuals so individuals with severe disease of SARS-CoV-2, some proportion of them actually were positive for Candida species, suggesting that IL-7 active antifungal or IL-17 signaling at the time of infection may be detrimental to good outcome for severe disease. And so that has really sort of these kinds of things have helped motivate one of the more important questions I've been seeking to answer is how does nasal state impact infection trajectory? And so often we think about immune cells as being the main effector responses in a viral infection. But I want to make the case that when we're thinking about infection in the tissue level, we really need to be thinking about the entirety of the tissue. And so when we think about a tissue microenvironment like the nasal mucosa, not only are we thinking about the immune cells that can infiltrate this tissue, like dendritic cells, monocytes, T cells, we also have to be thinking about our resident cells, which might be resident immune cells, as well as epithelial cells and some fibroblasts. And finally, we also need to be thinking about permanent resident species, things like sensory neurons, endothelial cells, and plasma cells, which might be there for nearly the lifetime of the organism. And I want to propose that really all cell types and cell subsets have the capacity to initiate, sustain, and maybe even remember inflammation, and therefore participate in a viral infection and provide some amount of protection or susceptibility. And so the general hypothesis that I've been very interested in pursuing is that the quality and timing of the multicellular responses during a viral infection determine the disease outcome, not just necessarily the amount of antibody present or the levels of the CD8 T cells you have in that tissue. And so in order to try and get at this and really understand what's happening in the nasal mucosa, we took a unsupervised approach to study this tissue in a mouse model. And so here, working with a graduate student, Colette, in the Von Andrian lab, is we have a mouse model of influenza infection restricted to the upper respiratory tract of mice. And what we did is perform single-cell RNA sequencing longitudinally across the acute infection trajectory, sampling 3 different regions of the nasal mucosa. Because of time today, I'm not going to be able to explain really the caveats of all these different regions, but suffices to say that the murine nasal mucosa is just as complex as the human, if not more so, given the necessity of different functions that this tissue plays. And so we sought out to answer this hypothesis that test this hypothesis that viral infection induces tissue scale changes to cellular composition and phenotype. And so looking at the 10,000-foot view of the data set, Here I'm showing you a UMAP projection of our single cell data, where each dot is a single cell for which we have a cellular transcriptome. And here I'm showing you data from 5 time points, 3 different tissue regions, and 3 replicates, so 45 samples. We captured about 156,000 single cell transcriptomes, and we're able to pick up a wide variety of cell types that make up this tissue, including neurons, mostly olfactory sensory neurons, a variety of immune cells, a variety of epithelial cell subsets, fibroblasts, endothelial cells, and other stroma. If we look at how the different cells we captured come from different tissues here, green being the respiratory mucosa, purple being the olfactory mucosa, and yellow to orange being the lateral nasal gland, what we see is that the makeup of the different regions is not exactly the same. It's a very diverse and complex tissue where a lot of olfactory sensory neurons are captured from samples coming from the olfactory mucosa, granulocytes and hematopoietic stem cells as well, due to the amount of bone marrow in these tissues, whereas the respiratory mucosa has much higher proportions of epithelial cells, myeloid cells, and T and NK cells as well. And so this is the high level picture. What if we look a little bit more closely? And what I just want you to get away from this slide is that we're able to pick up quite a variety of immune, epithelial, fibroblast, stromal, neuronal subsets in this complex tissue microenvironment. And we can understand how some of these are relatively distributed in terms of cell numbers and where they exist. And because of the time I have today and just the magnitude of the data, I'm not going to be able to tell you about all the different subsets of cells, but we captured over 127 clusters. And this is one way that we can display the complexity of the data. So how does an infection unfold in this tissue? Well, first we can look for cells that have influenza transcripts and see that mostly epithelial and myeloid cells are exhibiting influenza transcripts. We can look where in what tissue regions we're capturing cells with transcripts. And so we see mostly these in the respiratory mucosa, along with 2, 5, and 8 day post-infection, and also a few cells in the lateral nasal gland. We can also look at the broad interferon response dynamics in this tissue and see that while type 1 interferons turn on in a bulk tissue setting at 2 and 5 days post-infection, we don't actually start seeing responses to interferon until 5 days post-infection, suggesting some sort of restriction, at least at the time point we measured, of host response mechanisms. And finally, we can image the tissue itself to try and identify where the influenza is specifically by staining for influenza protein and finding it along the sort of ciliated cells along the septum and the turbinates. And so I'm going to summarize a lot of data now on the next slide just to talk about the immune cell responses. So if we break down the infection trajectory into these time points, I'm going to show you some of the major immune cell responses that occurs at each of these time points. And so first, at 2 days post-infection, the only immune responses we really see are neutrophils. And so we see the accumulation of these mature and interferon-stimulated neutrophils that start at day 2 and then are sort of maintained long-term during the infection trajectory. And so here, each dot is a mouse replicate. And I'm showing you the proportion of cells of all neutrophils, for example, that are coming from these different clusters. At 5 days post-infection, we see the emergence of a huge proportion of interferon-stimulated monocytes and these CCR7 positive dendritic cells, as well as the appearance of interferon-stimulated T cells and cytotoxic NK cell responses. At 8 days post-infection, the monocytes now give rise to these interferon-stimulated monocyte-derived macrophages. We also start to see our T cell responses appear with our CD8 effector T cells and our Th1 CD4 T cells. And finally, at 14 days post-infection, we see our tissue resident memory like T cells start to appear, and also IgA producing cells as we start to have cells in the tissue that can produce antibody. And so now I'm going to focus the rest of the talk really talking about epithelial cells, which have become more of an interest of mine and something I think that's really interesting in this tissue given its diversity. And so here I'm showing you all of the different epithelial cell clusters in the data set. And I just want to highlight that we see a variety of different types of epithelial cells. And I won't take you through everything, but we're able to find basal cells, the stem cells of this tissue. These cells can differentiate into goblet and secretory cells, as well as ciliated cells. We find ionocytes and tough cells, which are more recently described in the airway, as well as sustentacular cells, which provide support for olfactory sensory neurons, as well as some rare subsets of cells like these keratin 13, MEG3, and KLK1 positive. And so first I wanted to ask the question, which epithelial cell subsets change during infection? Very quickly, we saw that the strongest changes in terms of proportion were in our interferon stimulated epithelial cells, which makes sense. A lot of the cells that were harboring viral reeds fell into this cluster. What we also saw is that cycling basal cells also increased in proportion at 5 days post-infection, which suggests that there's some amount of tissue repair. We were particularly interested in the cycling basal cells because other studies in SARS-CoV-2 and other infections have shown that in the lung, cell cycle or proliferation and interferon responsiveness are non-compatible. If you knock out interferon receptor, type 1 or type 3 interferon receptor, you have enhanced proliferation of basal cells in the lungs. What we were actually able to see is that there's no association, negative or positive, between the amount of interferon responsiveness here on the y-axis and the amount of cell cycle or proliferation here on the x-axis, suggesting that maybe in the nasal mucosa, these programs are not mutually exclusive and can be existing together. So if we take a little bit closer look at the epithelial cells, we're also able to see is this rare population of keratin 13 positive cells. We're particularly interested in these cells because they appeared at 14 days post-infection following resolution and expressed several markers that suggest they're communicating with immune cells like TNF, IL-1 alpha, interferon gamma receptor. And so we were able to image and identify these cells using keratin 13 by immunofluorescence and finding them in the anterior nasal mucosa here in green and along the nasal floor here, suggesting also their localization, and keratin 13 suggests they're squamous. If we look at the proportion of different cell subsets over time, we can see is that at 14 days post-infection, when we have our highest amounts of our TRM cells and CD4 cells, we also see increased amounts of these keratin 13 positive cells. And we were surprised, or I think encouraged to see, is that these keratin 13 cells express CXCL16, while the TRMs express CXCR6. And so we wanted to validate that this potential interaction was occurring on the nasal floor. And so we took RNA scope approaches here to visualize these keratin 13 positive cells using white for keratin 13 and CXCL16 here in green. And we see nearby CXCR6 expressing cells, suggesting that this is a putative interaction here between these epithelial cells and T cells in the tissue. And we now are working with a model to try and more specifically identify and perturb these cells in vivo. Finally, we were interested in seeing if CXCR6 was actually important for tissue resident memory T cell function and protection. And so we have the CXCR6 knockout mice, and what we see is that in primary infection, CXCR6 knockout doesn't seem to make a difference in controlling infection. But if we look at a re-challenge setting where we don't have antibody mediated protection in a heterologous setting, sort of like what Matteo talked about earlier, we can see that in knockout mice for CXCR6, there's reduced control immediately upon reinfection, suggesting that CXCR6 may be important for the maintenance of tissue-resident memory cells in the memory phase. And so I presented a lot of data very quickly. How do we summarize all this information? Well, what we can do is start to do a compositional approach where we, instead of measuring genes and cells, we can look at each dot here being a sample and understanding how the samples are related to each other based on the abundances of the different cell clusters that we measured. And so here, the respiratory mucosa, olfactory mucosa, and loudonasal gland are quite distinct from each other. We can understand how these different subsets of cells are related by looking at the weights of this principal component analysis. But I just want to focus in on the respiratory mucosa specifically because what we saw in the respiratory mucosa where infection was mostly happening is this really beautiful stepwise response to infection from naive to 5, 8, and 14 days post-infection. And because this is a principal component space, we can identify different clusters that are associated with different time points, like resting basal cells in our naive samples, cycling basal cells at 5 days, effector CD8 T cells and PDCs at 8 days, and our adaptive immune cells coming up at 14 days. And so here I've shown you that this is mostly a stepwise response to infection, but if we want to build better vaccines and really understand memory, we need to do a memory experiment. And so what we did is we did a memory experiment. We re-challenged mice with the same strain of influenza and then measured responses at 62 and 5 days. So 2 and 5 days post re-challenge. And what we can do is compare the primary infection secondary challenge setting and see here that neutrophils sort of appear at the same rate in the secondary challenge here in purple, whereas we see a much faster rate of response for interferon-stimulated monocyte-derived macrophages. Our CD8 and CD4 T cells also respond very quickly, but there's no response in cycling basal cells, suggesting very little epithelial cell damage. So that really suggests to us that infection in the secondary memory really is characterized by induction of multiple immune cell responses simultaneously without epithelial cell damage. And so we can summarize this now looking at the same principal component approach. In green here is the response trajectory in a primary infection, and in blue is the response trajectory in a memory response, suggesting a smaller overall tissue change in response. We can quantify this. I know I'm running out of time. And so the take home really is an effective memory response allows the tissue to travel through a shorter distance in response. So I'll end there by just saying that we can now sort of start to understand what happens in an ideal memory response. And by really starting to derive what happens in ideal memory, maybe we can start to figure out what the key components are of primary infection that establish this tissue scale memory in a re-challenge setting. And so I'd just like to end there and thank Erica, who's a technician in the lab who helped with the RNA scope data, and Colette, who helped develop this model. And I know I'm a little over time, so if there is any time, I'll take a couple questions. Thank you.
Thank you, Dr. Kazer. Really, really incredible data. Thank you for sharing that with us. We do have some questions in the chat, and I think we'll go ahead and go through them. So you mentioned interferon-mediated immune cell stimulation. How about direct antiviral effects of interferon on different non-immune cells? Did you look at interferon-sensitive genes that were activated?
Yeah, so we do see interferon responsiveness on many of the different cell subsets in the data set, so epithelial cells, fibroblasts, endothelial cells. The flavor of response is different depending on the cell type. And so we see certain sort of innate restriction factors like different IFIs and IFITs that are specific for different types of cells, as well as the GBPs are varied depending on the cell subsets. But the large interferon wave really occurs at 5 days post-infection. And then the gamma wave, we believe, is occurring at 8 days post-infection based on the presence of Th1-like cells and also we're able to measure interferon gamma by qPCR.
Thank you. Next question, any insight into CCL17 and CCL20 expression in the homeostasis to infectious state shift?
That's a great question. I haven't looked at those specifically. I saw that that's Payam's question, so I'll follow up with you, Payam, since I know we've talked about this data before.
And one more question from Payam. What influences tissue anchoring in the nasal mucosa?
I'm a little bit confused by the question. Can you rephrase it?
I can, yeah, absolutely. I'm thinking more about, you gave a really nice insight about what influences different immune cells to home to the tissue. But Dana Farber, for example, talked about the kind of don't leave signals that occur afterwards and the specific localization.
Yeah, so we know for T cells in other tissues that CXCR6, CXCL16 is important. And hopefully I showed a little bit earlier that we do think that plays a role in maintaining the tissue resident memory T cell population here. We also know that CXCR3, CXCL9 is critical. And while we don't see extended expression of CXCL9 and CXCL10, that's very easily turned on by gamma. There are likely other cytokines or interleukins that play an important role here, but we haven't dug into that. And I think I need to do a better job of really looking at the difference between day 60 and day 14 and naive. And that'll give us a better idea of like what are the maintenance signals in the tissue. Thanks for the question.
Thank you, Sam.
Okay, thank you so much again for